Okay, well, welcome. My name is Troy Evans. I am a current co-chair of UDN's PEER. And for those unfamiliar, the UDN is the Undiagnosed Diseases Network. Currently consists of 12 clinical sites across the country, a sequencing core, 2 model organisms screening centers, a metabolomics core, and a central biorepository. PEER is the Participant Engagement and Empowerment Resource. It's made up of patients and families who have participated in the UDN. The PEER members work with UDN researchers to improve the participant experience, connect families with each other, and share the UDN with others. Today, we are thrilled and extremely fortunate to not only have Dr. Euan Ashley fighting for us in the rare and undiagnosed community, but also to hear from him on this call. Moderating this discussion and introducing Dr. Ashley is our fearless peer leader and my co-chair, Megan Halley. Megan, please take it away.
Thank you so much, Troy, and welcome everyone, and welcome Dr. Ashley. I just would echo everything that Troy just said. We feel so fortunate to have you here with us today. And it's absolutely my honor to introduce you as a co-chair of PEER. And also as your colleague at Stanford University, though I'm in the Center for Biomedical Ethics, as well as my role on PEER. So for those of you who don't know Dr. Ashley, he hails from Scotland, where he completed his medical degree at the University of Glasgow, followed by a medical residency and a PhD in molecular physiology at the University of Oxford. He then moved to Stanford University, where he trained in cardiology and advanced heart failure, joining the faculty in 2006. His group focuses on the science of precision medicine, and as you can read about in the Genome Odyssey, in 2010, he and his team carried out the first clinical interpretation of a human genome. Since that time, the team extended this approach, and they now routinely apply genome sequencing to the diagnosis of patients at Stanford Hospital. And that includes, of course, our UDN group, and Dr. Ashley is one of the principal investigators for the Stanford site of the Undiagnosed Diseases Network. He's received multiple awards and honors for his work, advancing clinical genomics and precision medicine from no less than the Obama White House itself. And as if that were not enough, he is also the father of 3. I now know he plays the saxophone, which I did not previously know, and he's also generous enough to serve as a mentor for me on my own research on the ethical issues related to new genomic technologies. So with that, I'd like to welcome you, Dr. Ashley, to our webinar today, and thank you so much for joining us. I really wish that we could have this conversation in person, to do it with a glass of scotch, which I understand you are an avid student of its health benefits, would be fine with me as well, though I might choose bourbon since I'm American, but we can disagree to disagree.
We could talk about that, yeah.
So I hope that this can be an informal conversation and really just a chance to learn more about you and what led you to write this book, and where you see things going from here for patients and families who are trying to understand the role of genetics in their lives and the lives of their loved ones. So before we get started, just a couple of housekeeping items. So for everyone who's joined us today, please post your questions to Dr. Ashley using the Q&A function of the meeting, not the chat. So I'll be taking questions in the Q&A function. And I also believe you should be able to upvote questions. So if you read someone else's question and you would like Dr. Ashley to answer that as well, you can select it, and that will kind of move it to the top of the list. I would also appreciate, if you're willing, tell us a little bit about who you are and what brings you to this topic with your question. There are going to be a diverse group of folks in this room, and it would be great to know who else is in this virtual room with us at least. And I'll do my best to convey everyone's questions and to get to everyone's questions as I can, though I may combine questions if necessary for time. But as the moderator, I'm going to take the moderator's prerogative and ask the first question. So Dr. Ashley, in your introduction, it seems like you already have a lot of jobs. I'm curious, why did you think you needed to add author to that list?
That's a great question. And let me start by saying, though, first of all, thank you both, Troy and Megan, for the very kind introduction and for having me here. I have to say that, you know, as you'll hear, and I promise to answer your question, that I wrote the book because I was just in awe of the stories and the narratives that our patients are living through. And I think that I wanted to try to spread that word. And so I'm particularly happy to be here today, and we're doing a few different launch events for the book. But in so many ways, this is clearly the most unique one, because this is the one that speaks to the people who are really at the core of the book. And the book tells scientific stories, and it tells stories of new technology. But the most important stories that are really the beginning, middle, and end of the book are those around patients with rare and undiagnosed disease. And so that's why I'm really just delighted to be here today, and can't think of a better event to have to try and speak to the people who I really was writing about. And so, yeah, to your question, you're certainly right. My wife would agree with you. I already have too many jobs. But writing is one of those things, first of all, that I came to science through writing. I would say that, first of all, and that as it turned out, many people are inspired by a special teacher that they had perhaps at school. It turned out my high school teachers were not that inspiring for science, it turns out. And I learned virtually nothing in my biology class. But my biology teacher was, interestingly, a saxophone player. And so I used to talk to him about saxophones and jazz after school, after class. But he did something which was really ended up being formative for me, which is, although he didn't really teach me anything in the class because he had no control of the class, he gave me a book to read one day and said, "You should read this. I think you might find it interesting." And that book was The Selfish Gene by Richard Dawkins, a very famous book about genes and genetics. And in fact, for many people might know the word meme these days because memes are everywhere on social media. Richard Dawkins, in that book in 1976, that was the first time he coined the phrase meme. So it comes from all the way back there. The last chapter of that book talks about a cultural unit of evolution and selective evolution. So we could talk about that another time, but he gave me that book. And I was just amazed at the clarity of writing and the explanations of science and the wonder of the biology of the modern world, the biological and natural world in this book. And at that point, I think the die was cast that one day I would try to write a book. I think, as I said, probably in maybe the acknowledgements of the book, I don't think my wife, she also knew that, but I don't think she thought it was going to be when we had 3 youngish children and both had full-time jobs. On the other hand, sometimes you just have to do it. And I think I've been overwhelmed by the stories of the lives of our patients. And I think the potential impact of the genome on those lives, and wanted to spread the word a bit further. And I can even tell you, just to one little last story on why I decided to do it. I sort of thought, I'll do this when I'm retired or emeritus. That's what happens, in our world in academia. You become emeritus as a professor and slowly fade into the background. I thought that's a perfect time to write a book. But I was lucky enough, as you mentioned in your introduction, to spend a bit of time with the Obama administration and the Science and Technology Policy Group there. And they were really driven by Obama's own love of science and technology. And I was at an event, very lucky to be an event in Pittsburgh that had been put together near maybe the last year of the Obama administration. And I was in the audience, and Atul Gawande, who's a doctor, a physician, and author who I love, was interviewing Obama, who turned out was a big technology geek. And they were talking about science and technology and its importance, and also of how that sort of technology could have an impact on medicine and on patients' lives. And I just remember sitting in the audience and watching these 2 amazing people and thinking, both amazing writers individually as well, I shouldn't wait. I should just do this. And so on the way back on the plane from Pittsburgh, I started writing, not without really any sense of whether it would ever become a book, but started to tell a few stories from my patients. In the end, it turned into a book. And I'm so happy that the stories are out there. I'm a bit nervous, as a non-author, still don't quite believe I'm an author, but I guess there's a book. So at some level I must be, but anyway, so yeah, that's how I kind of fell into it, like so many people, rather than it being, at some point it was in my destiny, but the actual timing of it was kind of inspired by that.
Yeah, the timing of it is really interesting. I must say, I didn't realize until I got to the close to the end of the book that I was really reading a history book in some ways, but really a history of the birth of clinical genomics, but told through these very, very personal stories. And I particularly appreciated the tone. You know, I've joked to you that it's just the right level of information for a medical anthropologist like myself to understand. You know, it's not too heavy on the science, but it really, I think, strikes a balance between providing enough detail to really understand what's so innovative about what you're doing and what your team is doing, but also being accessible. And so you know, we're really in an age where science communication and understanding science is at a tough place in this country. And so I'm curious how you've thought about and you know, really how you, I think, succeeded in really striking that balance between, you know, including the details to really show what's incredible about what you're doing and also making it accessible. I think that's a real challenge.
Well, it certainly is, and you're very kind to say that. And I certainly I tried hard. I grew up, as I mentioned, with this love of science that mostly came from reading popular science books that were designed for regular people. And that was where I got my first love of science before I ever ran any science experiments anywhere. And so having been really impressed with those authors, I was hopeful to include some science, because I think that at some level, while the human narratives and the stories of the families and patients are really the central thread and the central core for the book, I think people also learn to read to learn new things, and they like to be taught new things. Actually, it was an interesting quote. I don't know if it made it into the book, but when I talked to Francis Collins, ahead of the NIH, as many of you know, about his time with President Obama, one of the comments he made was that he was so impressed with the former president, and one of the things that really impressed him was that no matter what was going on next door, and it could be some global crisis, he always was really interested and present, and really wanted to be taught something, which I thought was interesting, that he wanted to learn. He wanted Francis Collins to teach him about not just about patients and to hear the stories, but also the science that underlay it. And so my hope was to not to lose people, but neither to talk down to them, because I really do believe, although there are some complex aspects of interpreting a genome, that at the end of the day, it's the centerpiece of our own biology. So we should be able to understand it, and that if it's explained well, then even complicated things make sense. I mean, one of the stories in the book I talk about these two little boys, Carson and Chase, and you know, at the end of the day, we did a sequencing of the family, Danny and Nikki, the mom and dad, and the two boys, and we were able to find an answer, as we do in many of the cases with the UDN, by sequencing all four of them together. And it turned out that the answer was a deep little known metabolic pathway in mitochondria, the powerhouses of the cell. So at some level, that's very complicated, and if we were to write it out, it would look very complicated on paper. But on the other hand, I think we all understand energy, energy in and energy out. We understand our cells are driven by batteries. And in the end, I tried to find that metaphors and analogies to try and help explain what might be complicated biological processes, but in reality are not so different from things we see in the world. And in the end, I chose a little factory of doll making for the metabolic pathway there. And the idea that if someone is not on the factory line that day, then a few different things happen. It's not just that the line stops. That, of course, happens. But the parts that were moving on their way down the line start to build up. And that can be a problem. And then, of course, you don't get anything downstream. And that's relevant for how you could treat, in this case, a rare disease caused by this deficit in this metabolic pathway, because you could jump in downstream and add in this case, like a fully clothed doll down the factory line, as one way of attacking aspects of the disease. Of course, with genetic therapy coming forward, we hope to be able to put in sort of exactly the right therapy at the right place at the right time, which should be a new worker who knows exactly what to do at that point in the factory line. So I tried to, if wherever possible, also to bring those kind of scientific analogies together, mostly because that's how I started learning science.
Yeah, absolutely. And I'd love to ask you a little bit more, to share a few more of your thoughts in that area in particular. You know, the book, most of the book is not entirely, but very focused on diagnostics and how you've really been part of pioneering this approach to diagnosing rare disease using genome sequencing. But you touch on this in the book, but I think it really comes through in the last chapter. You know, what comes next? You know, how do we move from, you know, diagnosing these rare, ultra rare diseases to thinking about therapy? And I wanted to share a question from one of our audience members, who's also a fellow peer member, Brian Broadbent. He's at the Baylor site of the Undiagnosed Diseases Network, and he has a beautiful little girl named Emma, who we've all been following her story very closely. And Brian is going to forgive me if I get some of these details wrong as I'm describing Emma, but I'd love to ask you this question, not necessarily specific to her case, but, you know, as an example for the audience, you know, how complex these diagnoses are, and then, you know, how you think about a next step. So Brian talked about Emma as the first person in the world with a genetic disease caused by INC RNA, a gene called chaser, and it regulates the CHD2 protein. And they were so excited to get this diagnosis, and they're still working on a paper, but they've really been struggling with where to go from here, and working with some really great scientists in their local area. But of course, things move slowly. You know, it's hard to, they don't even have another person, you know, to be working with, or another family. Now, how do you see, so both for Brian, but also for patients, like patients served by the UDN, you know, what is the model moving forward? How the more we identify these ultra rare diseases, the more diseases we have to treat, which is great and also complicated. And so, you know, what do you see as kind of the big picture policy things that need to happen? And what advice do you have for patients and families who might be at this point?
Well, yeah, there's a lot in there. Great questions, and thanks for sharing this story, Brian. And well, let me start by saying that if the last 10 years were very much about bringing the genome from a point of being able to just spell out the letters to the point where it could be a genuinely useful tool for patients really across the spectrum, but particularly in the rare disease space, I think the next 10 years are going to be about harnessing that information towards therapy. And I think some of that therapy will be genetic therapy, and that we will get orders of magnitude better at translating the diagnostic information into therapeutic technologies that can be applied even at an N of 1 or unique in the world frequency. And I think genomic sequencing will continue to improve. It'll get cheaper, it'll get faster, it'll get better. We'll cover more of the genome. And I'm excited about that. And for sure, we are working on that as the lab as we move forward. But what we've been spinning up in the last few years actually is a much more significant effort in genome engineering. And the idea basically of using tools like you might have heard of CRISPR gene editing. I'm sure many of who are listening have heard of that. It's been on the paper, also front pages of the newspapers, but also Nobel Prize this year for Jennifer Doudna and Emmanuel Charpentier. So the idea that you could potentially correct back to non-pathogenic changes and variants in the genome is incredibly exciting. And I do think that having this higher throughput technology, this cutting edge ability to read the genome, needs to translate to a rapid ability to write the genome and correct it. So it's obviously not going to happen immediately. But it also, and you mentioned this a few moments ago, that much of the book covers the last 10 years, which is in some ways the historical portion of us taking the genome from a bunch of letters to a tool that we can use for patients. And we're just at the beginning of the equivalent for genome engineering, which is to say that having the ability to understand what is wrong, which is ultimately what the Undiagnosed Diseases Network began with, has to then translate to what comes next. And so it also mirrors what the UDN is doing, which is increasingly paying attention to therapeutic possibilities that are brought up by the diagnostic potential. Now, it doesn't have to be genetic therapy. Of course, in some ways, the shortest distance from a diagnostic, a genetic diagnosis to a therapy is a genetic therapy. But of course, we haven't solved all the issues of how to get the right CRISPR or base editor into the right cell at the right place at the right time. And there's been amazing stories of early successes in that range. But I think if I heard correctly, this is a link RNA, a long non-coding RNA disease.
Yes.
Yeah. And so there are certainly RNA tools for manipulating RNA silencing. It was actually, even before CRISPR, there was a decade worth of interest and advancement on the idea of RNA silencing. And we've, in our lab, been using RNA silencing approaches to push forward treatments for cardiomyopathy, heart muscle disease. And now we're also moving towards this idea of word processing the genome, if you like, which is CRISPR gene editing or base editing. And so I do think that this next 10 years is going to be really important, and we're going to see really major and rapid changes in that space as we learn how to get these kind of gene editing tools into the right cells at the right time. So very excited by the future of that.
Mm-hmm, mm-hmm. Yes, it strikes me, you know, that the first stories you're telling in the book are barely 10 years old, yet so much has happened from, you know, talking about the story about Steve Quake and the first clinical interpretation to where we are now. It really has happened over what feels like an incredibly short period of time. And just to think, you may need to write another, your wife may be upset, you may need to write another book in 10 years.
Yeah, that's right.
Because there's going to, who knows where we'll be 10 years from now. I don't think 10 years ago we could have imagined where we are now. Maybe you could, but I certainly could not.
Well, no, I think that's a great point. Actually, I love this quote. I think I used at the beginning of the last chapter of the book, but from Bill Gates, who said, most people overestimate what they can do in 1 year. And that's certainly true, but they underestimate what they can do in 10. And I think that's true in the sense that when we first started working on Steve Quake's genome, we sort of had the sense that, oh, well, genomes are going to be everywhere. It's going to be so much cheaper and everyone's just going to have their genome sequence. Of course, that didn't exactly happen. And it took even a few years before we could get this kind of information even just available to patients who really, really needed it. And in that case, it was mostly exome, that 2% of the genome that codes for the genes. Still a very useful tool, but not quite the whole thing. But then when you look back, as you say, on the 10 years of where we came from, when we were, 10 years ago, just over 10 years, we were just prior to, say, the Steve Quake genome that starts the book, you were talking about $200,000 in months just to do one genome. And 5 years before then, it was $100 million to do that. So that level of change is really, at least when I look around at the history of medicine, I don't see examples of technological change at that speed of progress. And especially because it has spawned so many other things. I mean, everything, of course, from the diagnostic part that we're focused on here, the therapeutics that we just discussed, to also, let's face it, genomic vaccines is what's going to save us from the pandemic, essentially. Being able to sequence the SARS-CoV-2 virus is what allowed us to get testing rapidly and to have the fastest ever generation of these vaccines. So it has been an incredible change in technology over the course of just a short period of time.
Mm-hmm, mm-hmm, absolutely, absolutely. It was pretty mind-bending to read, to read what's happened in such a short period of time. Though I have to say, I was reflecting, reading the stories of these patients who you cared for, and your descriptions of them are so heartfelt, and you clearly are a clinician who cares deeply about his patients and their well-being, and are up late nights and weekends, trying to solve these puzzles. I suspect that for many on this call, and in some cases, in some points, in our journey with my son, who's an undiagnosed patient in the UDN, that was not our experience. We've had really a lot of challenges, trying to navigate the health system. And so I guess my question is, we've made the technological advances, but in many ways, our healthcare system still is not really designed to care for patients with these multiple complex overlapping issues that cross specialties that, and so, we have wonderful geneticists who diagnose, but they aren't really, well, at least not at this point, really involved in follow-up care. We have cardiologists like yourself who really learned and developed this expertise in the genome. How do we, as on a healthcare system level, how do we start to move towards a model where these types of technologies are more broadly available to patients?
Yeah, it's such a good question, isn't it? And your own story really informs your question there. And I feel so bad when I hear this story. So I mean, it's all pretty much universal, isn't it? I don't think that we have a patient who knocks on the door of the UDN who doesn't have some version of that story that they've lived through. And I think you put your finger right on it. Our healthcare system is just not set up for complicated, multi-system, unusual patients to walk in. When you think about a 12-minute primary care visit, it is possible to change a blood pressure medication in that time or decide to send off a cholesterol medication. But to get a handle on, a pile of papers this high from 14 different medical specialists, some of whom have some clues and some of whom have no idea what's going on, I think our system is just not set up for that. And it's actually a question that came up, interestingly, when we were putting together one of the first big papers, and there's been lots of papers, thankfully, from the UDN scientific papers, but I was lucky enough to help kind of pull together one of the summary papers. And actually, when we were going through the process of having it peer reviewed, which is when you have anonymous other scientists read the paper, give you feedback through the editor, and you improve the paper, you hope, and finally get it published. One of their questions to us that we were kind of pushed to answer was, this is all very well, the UDN, for this select group of people who are lucky enough to get it. They have doctors who have time and who have time to spend hours thinking about it, to talk to bioinformaticians and biologists and make cell lines from patients and all this. But what about everyone else? What about the people who don't make it into the UDN? And I think that's really important. And I talked a little bit in that response, and then there wasn't much room in the paper to say it, but I do think there's a real role for groups like the UDN creating essentially a trickle-down phenomenon. And that is kind of a loaded expression with economics, but I do think it helps explain. We need groups to be able to do everything that we have and understand how to apply the newest technology to the neediest of cases. But just as much as we spend time on that, I think we have to think then about the immediate next level down, which if it's a pyramid, that has to be a broader base. How do we trickle down to that next level where maybe not every single one of the newest tools is available, but the ones that we've proven to be useful and good, and in the case of the UDN, cost effective. I'm not sure if everybody on the call knows, but the analysis we did for that paper showed that if you brought the genome earlier, you could reduce costs by 96% to the healthcare system. Average costs were in the hundreds of thousands, and that doesn't even begin to touch on the emotional cost, but the financial burden was in the hundreds of thousands of dollars. And the UDN approach was more like in the lower tens of thousands of dollars. So it was a significant reduction in cost as well. So there is actually an incentive for the healthcare system to provide this kind of care. But the other challenge then, so if there's a financial incentive, that's good. But the other challenge is, where does it fit in? Because it doesn't fit into a 12-minute primary care visit. Clearly, geneticists, as you mentioned, are very well equipped to understand the genetic basis, and they do a lot of undiagnosed disease sleuthing. But that doesn't speak to the complexity of many of the patients who present and who then end up with maybe 5 doctors for their 4 different subsystems that are involved. Who then navigates that system? And I often, that word comes back to me a lot, because I think that what we need in many ways to help families and patients like yours and like others who are on the call here is a healthcare system navigator, someone who can help organize and take some of that burden off the patient and the family, because they already have a big enough burden dealing with the medical problem. Navigating the healthcare system shouldn't be an additional burden. And so I would like to see how groups like ours can help sort of trickle down these ideas to form a layer of support that isn't natural or obvious in our healthcare system, but that can actually be implemented And a lot of it is around this idea of navigation.
Mm-hmm. I absolutely agree. I think, you know, the diagnostic achievements of the UDN are impressive, but the kind of team science, you know, accomplishments are, I think, equally impressive and equally important in order for the accomplishments in diagnostics to actually make it into the broader healthcare space. So I have a little bit of maybe a simpler question for you.
Okay, good. That one wasn't easy.
I know, sorry. How do scientists get paid for this type of work? One of our participants is wanting to better understand, you know, you take care of patients, but you also do research. You know, can you explain a little bit about how this type of research gets funded and who pays your salary when you want to do it? Yeah.
Well, that's actually a really relevant question, because it's not, there isn't one answer to that, and there isn't one place to go. But and I talk a little bit in the book about when I first learned that there was going to be an Undiagnosed Diseases Network, actually from David Adams, who was part of the UDP, the Undiagnosed Diseases Program at the NIH, which was the only one of its kind at that time. And I describe a little bit my excitement at the idea that the NIH would be stepping in to basically fund a program that did what many of us went into medicine thinking we were going to do all the time, but actually don't do very often, which is to start with a difficult patient challenge, try and help the patient through our care and attention. And then if we don't find an answer, take it to the lab and work out what the answer is there, and hopefully at some point even get a therapy back. Because there was no obvious way of funding that kind of work before. The way standardized scientific funding works, you need a big program idea with 3 aims, and then you apply. It takes you 3 months to write it, and you get lots of, you know this well. you have to get advisors and a committee together, and then everybody writes letters, but everyone writes them for everyone else. And so it's a lot of work just to put that in. And then usually what happens is the first time you get feedback, and then you have to go back in, and most people eventually get some funding. And that kind of speed of movement of science is not really, not that speedy, frankly. It takes a long time. And so it's not a very good system for funding the kind of work that you need to do for these families and patients, where you don't necessarily need a lot of money to start with, but you need to be able to go and look at those cells, to be able to take each step as it goes along in an incremental way. So the UDN is really unique in its funding model for that. And we're actually at the point of really focusing on the next chapter for the UDN and what happens just by quirk of the way the UDN is funded through the Common Fund part of the NIH. It has a kind of 10-year limit for that level of funding. And so we need to look beyond now at different mechanisms for funding. And that's a challenge, because there aren't obvious and easy mechanisms to fund this kind of work. And what ends up happening, and this is what we did before there was a UDN, and I hope that we wouldn't have to go back to this, but it will always be part of it, which is to use just we call it soft money sometimes, and it's sometimes raised by foundations, sometimes it's given by philanthropists. It's the sort of money that a scientist gets as in gift form. We have a 5K walk run that we do every year and raise some money, and that goes to fund these little, you might call them micro odysseys, the little scientific odysseys that happen in the lab to try and follow up and get answers. But it is actually, it's a really good question, because there isn't an easy answer. So we're always on the hunt for more sources and more resources. But the NIH has been, I think, very generous in funding the UDN, and I'm hopeful they're going to continue to fund it, if not in exactly the same way they do now, but in a substantial way going forward.
Mm-hmm. I know. I thank you for addressing that, because I know that that's something that's on the minds of a lot of the current UDN patients and families. And for those who are on the call, the peer group this is something that peer group is working on very directly. And if you have additional questions, you're welcome to reach out to any one of us about that as well. So I have an interesting question from one of our members, one of our listeners. So he's asking, should we all be studying our own genomes like discussed in the book, or is this only a Silicon Valley thing?
That's a great question. Well, yeah, So, you know, I think this goes back to the point I made a few minutes ago. When we first started doing this, the cost was going down so rapidly with genomes. We really kind of felt that everybody would have their genome soon. And in fact, some of the story in the book is about how we ended up starting a company to deal with this, because we basically were getting so many people asking us, can you analyze my genome? Can you analyze my genome? And each one would take, you know, hundreds of hours to do. And we just couldn't do that in our labs. And so we ended up starting a company to do that. It turned out that there was only a fairly small, in the end, number of people who really were going out to sequence their own genome and then having a hard drive that they needed help with. And it was an unusual time. I included some stories in the book. I couldn't include them all, but there are some very household names who approached us with their genome around that time. And I have some stories that will probably never be told, but just if you think about Silicon Valley and technology and think of the sorts of CEOs that you hear about in the news, then assume a certain percentage of those approached us at the time with requests to sequence their genome and analyze it. There's a few stories in there. But I think that to the serious point, the fact that this technology and this data is available, I think, has been revolutionary within the rare disease world. And that happened quite quickly. It didn't happen immediately. It was probably 2 or 3 years after the early stories in the book that it really took off. But it is routine in the sense that it's available. You don't get it through your GP, but if you get to the right geneticist, for sure, you would have access to an exome or a genome here, and that's helpful. Of course, I get asked a lot as an adult cardiologist, as well as the undiagnosed patients and the mystery diseases. I also see, if you like, see inherited heart disease patients, and we talk about them in the book, and also some more, if you like, regular patients where we talk about blood pressure and cholesterol and so on. And those patients are also wondering, so when is the genome available to me? And that's the other, if you like, 9 out of 10 people. If 1 out of 10 have a rare disease, and that's as common as diabetes, it's a big group. But then there's still 9 out of 10 people who are not in that group. So when is the genome coming for them? And that's part of, I think, the question here. And so we have an exciting update there too. Actually, I've kind of been asked that question every year in the last 10 years. And each year I've sort of said, not quite, not quite, not quite. And actually this year, in fact, kind of last year, was the first time I really said, well, the time is now. And it's a very different kind of use of the genome. So whereas with rare diseases, where I think on this call familiar, you're sequencing the genome, covering each spot, all 6 billion, really deeply. So you can, at any given position in the genome, you can say exactly which letter or 2 letters there are. Well, there's another version where you cover each part of the genome just much more lightly, like once or twice, basically. And that still allows you to cover the whole genome in a way that can impact your prediction of the likelihood of you getting diseases like heart attack, high blood pressure, diabetes, cancer. And they're sometimes called polygenic risk scores. And polygenic just meaning many genes. And it is many genes, but it's really many variants. A lot of the variants are actually in between the genes. And just to give you an example of how far we've come, if I lined up the traditional risk factors for heart attack, there are things you'll recognize like cholesterol and diabetes and smoking, this kind of thing. Well, we can make a genetic score that is more powerful than any one of those at predicting your risk of having a heart attack. And that's in some ways not that surprising, given we know 50% of your risk for a heart attack is inherited, 50% nature, 50% nurture is about where it comes down. And we've mostly ignored the nature part. We ask all about the nurture, the smoking and exercise and salt in your diet and all of that. We sometimes ask about family history, but not that closely. So these polygenic risk scores are finally giving us a window into that. And not surprisingly, when we account for both nature and nurture, we're able to refine our estimates. And at the moment, for example, you get a cholesterol medication if your risk of a heart attack over 10 years is more than 7%. But you want to use the most accurate prediction model that you can for that 7%, if it's the difference between taking or not taking medication. If we incorporate the genome information, then that makes for a much better estimate. And we're actually rolling this out in primary care and preventive cardiology at Stanford this year. So after 10 years of being asked when it's coming, and even the first model back in 2009 described in the first chapter of the book, we're actually rolling out starting in a few months. And so really the genome is coming for everyone in a way that I think is going to democratize beyond Silicon Valley is the answer to the question.
Yes, great. Thank you. Thank you for that. And that's quite an exciting update. We'll all have to stay tuned to see how that goes. I just want to remind our listeners that you can put questions into the Q&A function, not into the chat, please, because I can't see them as easily. But I'm trying to organize questions in the Q&A. Oh, and we have actually a question that just popped in about risk scores. So I'm gonna follow on from our discussion. Kara Anderson, who's another one of our peer members for the UDN, says she thinks risk scores are extremely helpful, but risks will never be absolutely zero. As the availability of the human genome increases, is this going to create a large diagnostic burden for certain risks that are not an health concern? And I actually love this question as an ethicist, because, you know, we like to be the downers in the room. And we really like to say, well, what about the drawback, about these unintended consequences? So, I'm joking, but there are some, of course, always unintended consequences that we need to think about as we roll out these new technologies. So I'm curious, you know, you've done incredible things with the genome, but I think, you know, there are examples we've seen, you know, for example, in breast and ovarian cancer, where they're actually currently recommending using screening for fewer variants because women are making decisions based on information that's perhaps not complete. So I'm curious, you know, from your perspective, what are your concerns with something like a polygenic risk score, or how widespread use of the genome might create challenges in other ways that we might not anticipate?
Yeah, it's a really important discussion. And I say, first of all, I hope you noticed that the first thing we did after we took on Steve Quake's genome was talk to an ethicist. I hope you liked that. Did you notice that in the book?
And especially because it was Dr. Greeley, yes.
Yeah. So that was very important. And he was, as I described in that chapter, very important part of the early ideas around how we should go about it. So in many ways, the birth of our approach to clinical genomics was like hand in hand, I mean, literally hand in hand with our ethics colleagues. It wasn't that, you know, as I think you probably find often, you get asked after the fact to come and like comment on something that's been done. But we really felt it was important from the beginning to have both a genetic counselor and an ethicist in the form of Hank Greeley, like embedded in our team, very much part of the team and in the meetings. And so we've definitely used that as our philosophy going forward, and not least with the work you're doing yourself now. And I think it's been a great thing for Stanford and for the clinical genomics community. And so, yeah, then to questions that are perhaps not ones that we've dealt with yet, because they, you know, polygenic risk scores haven't been, I think, particularly clinically relevant to date, but they are now being so. And the new program that we have at Stanford also has close links with the ethics team. I think what I would say in the answer to that, and there are many ways in which genetic data is rightfully treated differently from other kinds of data. And because it's a signature, it's a unique signature that we all have inside us. So it's identifiable in a way that your, you know, cholesterol number maybe isn't. But in relation to polygenic risk scores and the general principle of common disease, it's actually is, I think, a little bit more like regular data, because at the end of the day, what happens is there's a sense, let's say if we stick with heart disease, that if we're able to predict your chance of having a heart attack, that's something that can inform whether you have a cholesterol medication or whether it's time to take a blood pressure drug. At the end of the day, the key information about that is actually our best prediction using all available data. That available data includes things like your blood pressure and, you know, whether you have diabetes and whether you smoke or not. And what we're doing is rolling into that a genetic component, because we know the disease has a genetic predisposition. And so in many ways, I think we're just making something that's happening anyway in a primary care clinic all over more accurate. And I think that if we're about to take actions based on the accuracy of our prediction, and another one, of course, is for breast cancer screening. If you have greater than 20% absolute risk, they'll suggest you have MRI-based screening rather than mammography-based screening, so a more intensive approach. Well, if you're going to make that decision as to whether you need the more intensive screening, you want to do it with the best available data. And our data is never perfect, but we do know that by incorporating this broader genetic data, also for breast cancer screening, that we're able to refine those estimates and just make better predictions. So I think that where we are clear that that's the case, and that's certainly the case, I believe, for heart disease and breast cancer and prostate cancer right now, and we're looking at others as we go down the line. I think that's the case there for now. We need to look at each situation carefully. I think one of the things that really was of some ethics concern early is that we would run these scores, you know, you'd get this and you suddenly have risk scores and everything. And that I think is much more problematic, not least because they aren't necessarily valid for everything. And we're just beginning to, I think, to work down the line in terms of the validity and we call it actionability of knowing these estimates. But also, the more tests you do, the more likely you are to have one be positive by chance alone. And so I think it's really important to think about the broader picture when we're incorporating this technology. And as the listener was saying, nobody's risk is zero. You're just by living in the world, you're at risk for some diseases. But I think the way to think of it is that you start by getting up in the morning not really knowing which of those diseases you're at risk for. And obviously the preventive treatments that you could, or the preventive actions you could take are going to be different depending on which ones. And so some people I think might be quite interested in which one of those various diseases they're more at risk for. And so therefore which preventive actions they would be best spending their time focusing on.
Mm-hmm. Yeah, thank you for that. I have an interesting question from one of our participants asking about the types of patients who are in the UDN. So I know we talk a lot about obviously the patients themselves, the adult patients themselves, as well as pediatric patients. There are a lot of pediatric patients who are undiagnosed with a rare disease. But our participants asking, are there families in the UDN that have a parent without a diagnosis as well? Do you know if the UDN is looking at families in that way? In addition to,
so if I understand right, that the concept is of a almost like an undiagnosed family where there's two people affected with a similar condition, and we kind of take on the family. Is that?
I believe that's what she's asking. Yes.
Yeah. Well, yes is the answer to that for sure. And that situation actually is an attractive one from the perspective of a group of medical detectives, because family-based information is always helpful. I mentioned the story earlier of Carson Chase, where having the family-based information that neither parent was affected greatly helps the modeling we do of the genomic data to understand what it might be that has affected both, in that case, two boys. But really any family member who seems or would appear to have a similar syndrome vastly increases our power for detection and discovery. Now, as it turns out, actually, the more remote the relative and the more clear the two conditions are the same, the more power we have. And that's because you share less DNA with a more remote relative. You know, if your first degree relative, you share about 50% of your DNA with them. And so if a brother, sister, mother, father, or kid has the same condition, you do get about 50% more power because 50% is not shared. So you can sort of rule out 50% of the genome. But if you have, let's say, a third cousin or someone who's much more remotely related, then the amount of DNA those two individuals share is much, much smaller, maybe even single-digit percentage. And in that case, you can throw out most of the genome and just focus on the shared part, which gives you much more power for discovery. So yeah, actually, from the idea of trying to solve medical mysteries, if two individuals in a family are affected and obviously biologically related, then that can provide a lot of power for us in discovery.
Mm-hmm. Great, thank you. So I'm gonna take, again, moderator's prerogative and ask you another personal question, which is about the patients and families who remain undiagnosed in UDN. What kinds of things do you think we're missing? You know, where are we going next? What's our next hope? You know, families like ours are really hanging a lot on you guys, and we know we've placed our hopes in a very in the right place. But I'm curious, you know, what do you see coming next? You talked about, you know, people ask you when's the genome ready for me? But that's a different question coming from an undiagnosed patient in the UDN. They've already had their genome, but the genome hasn't really come for them in the same way. So
yeah.
Reflections on that.
No, it's a really important question. And we spend a lot of time thinking about this question, or thinking about the answer to this question, because I, as much as anyone, celebrate the fact that a third of the time, a little bit above a third of the time, we're able to find a diagnosis. And of that, 80% of the time we make a diagnosis, that is an important action. Some of the time it's therapy, some of the time it's ending the diagnostic odyssey, some of the time it's for family planning. We should celebrate that, and we do. But at the same time as we celebrate that success, we can never forget that we're leaving 2/3 of our patients without an answer. And so we need to work harder. We need to do better. We need to get up earlier and think more. And we need to keep pushing the technology until we provide an answer for all of them. So where are we looking? That's really what your question is. Well, some of it is being able to look more broadly, accurately, and deeply into the genome. That was kind of our first place. If it really appears, and we believe in our heart of hearts this is a genetic diagnosis, but that our current tools haven't found it, then we have to think about what better tools for characterizing the genome do we have. And so we started, for example, to use long read technologies. You might have heard the standard approach to sequencing uses a technology from a company called Illumina. And actually, that technology was born in a pub in Cambridge in the UK. And that story I tell in the early part of the book. But it was that UK company was sold to this company, Illumina, based in San Diego. And that's the workhorse sequencing technology. It hasn't changed much it's got cheaper, but it hasn't changed much in its fundamental technology since they invented it. As a result, there are areas of the genome that are not actually quite finished. And in fact, I was at a virtual meeting yesterday where they were a consortium known as the telomere to telomere consortium, which just means end to end for chromosomes, are working on finishing off, polishing the last parts of the genome because there might well be answers hidden. And they talked about 90 megabases, like 90 million letters that's a lot that they've just added to the genome. So there's going to be some answers in that. These other approaches where we talk about long read, instead of spelling out a word that's about 100 base pairs, 100 letters at a time, which is how the Illumina technology works, we have a new technology that can look at these words that are more like 15,000 letters long. In the book, I use the analogy of a jigsaw. If you're trying to put a jigsaw together and you have 1,000 pieces, that's going to take you a while. But if you only have 10 pieces, well, that's your preschooler's jigsaw, and that's going to be a bit easier, judging by usually there's a piece missing, judging by my kids' jigsaws. But just leaving that aside, so those better technologies, I think, and it's particularly our ability with long read sequencing to pick up big changes in the genome. Sometimes those short reads, although they're amazing at picking up single letter changes, they're not so good at picking up like a 2,000 segment change. So the longer reads can help with that. So I do believe that, first of all, that technology will take us further. I think we'll start to use RNA where we look at the message and we find changes that were there all along, but we didn't know where to look. So the RNA will help us. I think we may start to find some answers in the epigenome, which is to say that the genetic data, the genome itself can actually be modified chemically in certain ways that mean genes open up and close down. And so I think some of that information is coming along from the new genetic sequencing technologies. And then there's going to be some situations where the answer is actually not in the genome, maybe. And I think the most common place that we'll look is in the immune system, actually. And we're all much more familiar with the immune system probably this year than we ever were before, as a result of headlines about viruses and T cells and B cells. And that's kind of everywhere right now because of the pandemic. But we've had an interest in actually many discussions at the UDN about how we start to formalize our tools for analyzing the immune system. And there's certainly some of the conditions that we've diagnosed, we've found the answer in the immune system, not in the genome. And the interesting thing about the immune system, and again, it's been in the news, but of course, we have B cells that make antibodies and T cells that can attack cells that are infected with viruses. Well, each of those two kinds of cells have this amazing ability to rearrange their own genomes in order to create the lock and key that allows them to attack the pathogens and neutralize them. And they do that by rearranging their genomes. And so we can actually use sequencing if we pull out those B cells and T cells and sequence the particular area that gets rearranged. We can use the genome sequencing of those specific immune cells to understand what it might be that they're attacking. And so autoimmunity, where the immune system turns on itself, or autoinflammation, or inflammatory conditions, I think many of our undiagnosed conditions might turn out the answer might be found there. So obviously you could understand, like I have another 45 minutes of places that we might want to look. I want, the message is those who remain undiagnosed are really close in our hearts, and are people that, and families that we spend a lot of time thinking about, and that it's really much of the focus of development is how do we do better for that group, and just new information every month on genetic information. That'd be the last thing I should say on that. But every month we just around the world learn new things about new genes and new places, and that by itself actually leads to a significant percentage of solved cases month on month, just because the world knows more about the genome. So stay hopeful. That's my response.
Absolutely. We don't have any other choice, right?
Right.
So we just have a couple more minutes, and I just want to, you touched on this a little bit, but I want to bring up one more question from the audience about the Diagnostic Odyssey and the length of time that it takes to diagnosis, even if it's not an ultra rare disease like maybe you're dealing with in UDN, it's still a rare disease. It's not well known to the medical community, to primary care providers who maybe aren't at large academic medical centers. What do you see as the main barriers to making clinical genome and exome sequencing more widely available with the explicit purpose of shortening the diagnostic odyssey for rare diseases?
Yeah, and this is a little bit of overlap with the question we had earlier, but I think we can definitely dig in on one specific point that we didn't make. It is about, and we were talking about this sort of idea of trickle down a little bit. We try to do things in the UDN, but make sure they're being passed to this much broader group. I think it's really important, and I think unfortunately it comes down to payers a lot of the time, because I think, I mean, it is true that we don't have as many geneticists as we need and genetic counselors. And so we do have to train more doctors and expand those specialties and expand the education of those going into those specialties. But at the end of the day, the real barrier is actually our healthcare system. And I know we talked about that earlier. And thankfully, things have moved a lot since where they were, but we're still at the situation where, you know, if even at the Stanford clinic, let's say if you go and you're going to have an exome sequencing, they'll send you away from the clinic so they can spend 3 weeks on the phone with the insurance company deciding if the insurance company are going to pay for it. And then if they do, you come back to the clinic and get your blood drawn, and then there's another couple of weeks. And then, I mean, that's just unacceptable. And it's such a contrast. I'm involved a bit now with in the pharmaceutical industry. I'm on the board of AstraZeneca, and I've been sort of seeing what happens on the therapeutic side, which is, and there's really large amounts of money in therapeutics, and things move quickly, partly because those companies are very good at lobbying for what they need. On the diagnostic side, we have this challenge that because each disease is sort of individually rare, and the diagnostic companies are individually not necessarily that powerful, they haven't been able to move the needle. And so we have this real contrast in this country between the real challenge to get a basic and clearly cost-effective and oftentimes life-saving diagnostic like an exome or a genome reimbursed, whereas we're getting, not always, but we are getting therapies that sometimes cost very large amounts of money reimbursed, and it's an unusual contrast. And I think we would like, if I could just have my druthers, I would tilt the balance a little bit so that we can make the diagnostic much more broadly available. And what a waste of time. Like, this is a waste of the patient's time, primarily the most important, and the family. They shouldn't have to wait. But it's also a waste. We have people employed full time doing nothing but sitting on the phone all day with insurance companies. And I've talked even to the executives at many of these companies, and they go, "Oh, we know that's terrible." And I'm like, "Well, I had to do something about it." Like, you know, you're going to say yes like 2/3 of the time. So why don't you either just say yes all the time, and we'll change the cost or the price structure a little bit so you can say yes all the time, which is what they should do, or come up with some other plan where there's some tick box, and if you tick the 4 boxes, you just get approved. There's this idea of like, it's the reason that in the US the administration component of our healthcare system is so high, like 20% of healthcare costs is is administration, and it's more like 3%, 2% in other countries. So sorry that's such a banal and uninspiring answer, but it's the real answer. To get this more available, we need the payers to stop messing around and just approve the test. And we're way past the point where the data clearly is supportive.
Mm-hmm. Well, I actually think that's a great place to leave it on that very strong statement that I think everyone on this call could not agree with more. So we're right at the hour, and I just want to thank you so much on behalf of the UDN peer group for taking the time to speak with us today. And for those of you on the call who have not read the book, it's very accessible. It's an incredibly inspiring tale with lots of really wonderful patient stories that will help you understand the last 10 years of the Genome Odyssey. So thank you so much, Dr. Ashley, and thank you to everyone for joining us today.
Thanks, Megan, for having me, and to you, Troy, for co-chairing the amazing work you do at Peer. And thanks to everyone for joining us today on the call. You really are in our hearts, and we hope to keep working hard for you all the time.
All right. Thank you so much.
Thank you, Euan.
You're welcome. Thanks.
Bye-bye.
Thanks, Dr. Ashley. Bye.